final presenter, we have Andrew Chung, who's an MD PhD candidate from Texas A&M University College of Medicine. He's in the laboratory of Dr. Jenny Chang at Houston Methodist Cancer Center. And he'll be discussing the abstract evaluating the efficacy of toci for a subset of triple-negative breast cancer that resists chemotherapy via an autocrine IL-1A mediated self-induction IL-6. Thank you very much.
All right. Thank you all for having me. I'm sorry the title was so long. I wish I could make it shorter. My name's Andrew. I'm in Dr. Jenny Chang's lab with the Houston Methodist Cancer Center. It's a pleasure to be presenting. So I recognize most of the audience are very well versed in triple negative breast cancer or TNBC. So just real quick, these cancers are low expression for hormone receptors and the epidermal growth factor receptor 2. In short, patients with TNBC, they typically do not do well with standard chemotherapy, and they have high rates of disease recurrence and disease, cancer metastasis. And this is highlighted by a consistent, very grim 5-year prognosis for patients with metastatic disease. So in order to actually provide clinical benefit for our TNBC patients, we really need to get a better understanding of the cellular biology of the TNBCs, because TNBCs is a very heterogeneous type of cancer. It's not just one uniform type of cancer cell. So in order to develop novel targeted therapies, we really need to understand the biology a lot better. Having said that, to accomplish this, I am presenting one background slide on previous work done by others, where essentially they obtained hundreds, about 500 different TNBC patient tumors and performed microarray studies, and taking the gene expression profiles. These TNBC tumors were grouped by k-mean clustering based on their differential gene expression into the 6 unique subtypes that you see here at the top. And what I'm showing are basically the canonical pathways that are predicted to be upregulated in 2 particular subtypes. For the sake of time, I will not actually be discussing all 6 subtypes that the authors found, rather just the 2 subtypes that's more relevant to my talk. The first subtype is the basal-like 1 or BL1 TNBCs, and basically they have a gene expression profile that's highly upregulated in cell cycle, cellular division. In contrast, another subtype, the mesenchymal stem-like or MSL TNBCs, they have gene expression profiles that are very similar to cancer stem cells, angiogenesis, epithelial mesenchymal transition. And one canonical pathway that I highlighted in this red box is an inflammatory pathway, the NF-kappa B pathway. I just want to point out, although the authors made this discovery, they did not discuss the relevance of inflammation to mesenchymal stem-like TNBCs, nor did they ever follow up on this. So this was just kind of left hanging right there. Now, to actually validate that the original author's subtyping method had actual any type of clinical relevance, what I'm showing here are 2 independent follow-up studies where both groups, basically following the original author's methodology, they subtyped their TNBC patients and then determined if there was any differential efficacy to conventional chemotherapy. And quite consistently, both groups found that the BL1 subtype, which again has canonical pathways in cellular division, cellular proliferation, patients with the BL1 tumors had the best pathological complete response after chemotherapy, whereas the MSL TNBC patients, they typically had one of the worst pathological complete response after chemo. So in assembling my graduate thesis project, given how the MSL TNBCs are chemo-resistant and they have this not well elucidated inflammatory pathway, the NF-kappa B pathway, I basically propose to investigate whether the MSL TNBCs may utilize inflammation or autocrine cytokine signaling to resist chemotherapy. And in contrast, I used the chemosensitive BL1 TNBCs, which lack any inflammatory gene signature, therefore they won't be able to resist chemotherapy via any type of inflammation. Okay, so switching over to my data, basically what I'm showing here is I took 4 different MSL human TNBC cell lines and 4 different basal human TNBC cell lines and performed RNA sequencing analysis. And I analyzed the gene expression signatures that are up in the MSL TNBCs compared to basal TNBCs. And then I uploaded it to an online software tool called Ingenuity Pathway Analysis. Basically, I asked Ingenuity, could it predict for me of the gene expression profile signature that is up in mesenchymals? Are any of those, is any of that transcriptome data could any of those be an upstream regulator for that gene signature in MSLs? And what I highlighted here is basically IPA predicts that the IL-1 cytokine is predicted to be an upstream regulator in the MSL TNBCs. And as you can see, both IL-1 beta and IL-1 alpha, the red arrow indicates that the gene expression transcriptome data is indeed up in MSLs compared to basals. Now, for the sake of time, I'm going to just skip right ahead to my confirmatory study where I actually validate the RNA sequencing. So before I do that, I just want to explain real quick what docetaxel is. Docetaxel is a member of the taxane family, and docetaxel and taxanes are one of the frontline choices that's given to TNBC patients in the clinic. So for both my in vitro and in vivo models, I'm using docetaxel. And as you can see with the MSL TNBCs that are highlighted in red, in response to docetaxel therapy in vitro, they produce inflammatory mediators after therapy, after chemotherapy. IL-6 goes up, IL-1 alpha goes up, and COX-2 goes up. In stark contrast, this phenomenon does not really occur in the basal TNBCs highlighted in green. They make no IL-1 alpha. COX-2 really does not change afterwards. And in regards to IL-6, it's either inconsistent production or very modest production of IL-6 in respect to their MSL TNBC counterparts. So here, after establishing, okay, after chemotherapy, mesenchymals, they make more inflammatory mediators. Thinking back to the RNA sequencing data I showed previously, where the software told me that IL-1 alpha could potentially be an upstream regulator, this is where I test if that is true. So taking 2 different MSL TNBCs that were treated either with docetaxel alone, docetaxel with a irrelevant isotype control antibody, or docetaxel with a IL-1 alpha neutralizing antibody, neutralizing IL-1 alpha led to significant reduction of IL-6 following docetaxel therapy in these 2 cell lines. Likewise, you see that same trend with prostaglandin E2 production, which is the main metabolite produced by COX-2. And again, you see the same trend with IL-8. The reason I looked at IL-8 is TNBCs often, it's a report that TNBCs produce IL-8 in conjunction with IL-6 for their tumor resistance mechanisms. That's why I looked at IL-8. Now, previously I showed that the basal TNBCs, they produce no IL-1 alpha following docetaxel therapy. So I raised the question, okay, if I were to give them IL-1 alpha, is that sufficient to make them more inflammatory? And the answer is yes. Looking at 2 different basal TNBCs, simply just by treating them with recombinant human IL-1 alpha is sufficient to induce IL-6 production. Likewise, is sufficient to induce IL-8 production. So just to recap very briefly, the mesenchymal TNBCs appear to, in response to docetaxel therapy, initiate an inflammatory cascade where IL-1 alpha is the upstream cytokine that's responsible for all this. Now in this slide, I'm basically investigating what is actually the signaling pathway that's responsible for producing IL-1 alpha in mesenchymal TNBCs in the first place. After ruling out it was not due to NF-kappa B or AP1 activation, I discovered that the pathway is by the MAP kinase pathway, which was actually surprising to me since MAP kinase pathway is typically thought to, promote cellular proliferation. But in fact, what showing the data right here, docetaxel will actually lead to activation of MEK1 and 2, which will in turn phosphorylate and activate ERK1 and 2. And this in turn will activate a transcription factor that will lead to production of IL-1 alpha and so forth, inflammation. So what I'm showing here is PD-03 is a MEK inhibitor. So if I'm taking out this step right here, again with the MEK inhibitor, in the presence of docetaxel or even in the absence of docetaxel, by shutting this down, this will lead to a significant reduction of IL-1 alpha, again in 2 different MSL TNBCs. And by shutting down IL-1 alpha, it likewise leads to reduction of the more downstream cytokine IL-6. And this is just a Western blot to confirm that by shutting down MEK1 and 2, I am indeed preventing the phosphorylation and activation of ERK1 and 2. So again, the MEK inhibitor in the presence of docetaxel or absence of docetaxel will lead to decreased phosphorylation and activation of ERK1 and 2 in the 2 different MSL TNBCs. Okay, so in summary, what I found is, okay, great. Mesenchymal TNBCs, after docetaxel therapy, they'll have hyperactive MAP kinase pathway activation and lead to inflammation. How can I actually make this relevant for TNBC patients? Well, so one of the inflammatory mediators that mesenchymal TNBCs produce is IL-6, and IL-6 is very well studied, not only in TNBCs, but in all cancer biology. IL-6 is implicated to promote cancer cell proliferation, cancer stem cell induction, cancer metastasis. So in designing this project, targeting IL-6 in mesenchymals seemed like an ideal targeted therapy approach. So to do this, I obtained tocilizumab with the trade name Actemra, which is FDA approved for various arthritis pathologies, and is a humanized anti-IL-6 receptor antibody. And to investigate the efficacy of supplementing tocilizumab to docetaxel therapy, what I did was basically implanted non-obese SCID IL-2 gamma receptor deficient female mice, abbreviated as NSGs. Basically, these NSG females are severely immunocompromised, which will allow for successful implantation of human cell line xenografts. So in this experiment, the NSG females were implanted with a mesenchymal TNBC cell line, and in this experiment, NSGs were implanted with a basal TNBC cell line. And they were both treated and split up into 4 different groups, and as with this regimen shown here in the legend. For the MSL in vivo efficacy study, the 2 main groups of interest are red and blue. Basically, red is the group that is receiving docetaxel or chemotherapy alone. And when you compare that to the group that received tocilizumab, that's the anti-IL-6 receptor antibody, together with docetaxel, this led to an increased 2-week median survival with the combination therapy group. And this therapeutic benefit was not apparent whatsoever in the basal TNBC experiment, because essentially chemotherapy alone is sufficient to keep the tumor in check. In some case, leads to entire tumor eradication. So what I'm hoping to accomplish is basically one day we can identify a cohort of TNBC patients that may be ideal candidates of targeting IL-6 while they're undergoing chemotherapy. I'm absolutely not advocating we give tocilizumab to all TNBC patients, because that's simply not true. Not all TNBC patients will produce IL-6 as a resistance mechanism. The goal is to determine which TNBC patients will actually produce IL-6 after chemotherapy, and then supplementing those patients with targeted IL-6 therapy, such as tocilizumab. So this is just a summary slide of the pathway I elucidated. Again, mesenchymals after docetaxel therapy will have hyperactive MAP kinase pathway activation, which will lead to IL-1 alpha production, which will trigger a inflammatory cascade. And hopefully one day we can translate this over to TNBC patients in the clinic. I would like to thank Dr. Chang for having me as her graduate student over the years and for her continued support, and of course my lab. I could not be here without them, as well as my MD PhD program leadership and my collaborators. Thank you again so much.
Thank you. One question,
Yes.
It's certainly provocative findings. For the identification of the MSL subgroup, theoretically, if this pans out down the road, are there serum markers, or eventually that's going to be gene expression profiling on the tumor biopsy?
I'm very glad you asked that, Dr. Bernecker. So one could essentially, after tumor biopsy, perform either RNA sequencing. Now, in terms of how feasible that is in terms of cost and time for the TNBC patients, that's a little up in the air. What could be faster is basically taking the gene expression signature that is up in MS cells and just doing a qPCR profile, which it will be a lot quicker and faster and cheaper for those MSL target genes from tumor biopsies in order to make that determination, are these patients MSLs?
Interesting. Great, very good. Thank you very much.
Thank you.
That was great. You bet.